Very good. Thank you, those of you who come today. My name is Sam McKee. I am the Associate Tutor in History and Philosophy of Science at Manchester Metropolitan University, and I'm a DNA repair scientist at the University of Reading. And I'm talking to you today here at the International Society for Science and Religion Conference on the question of SynBio, synthetic biology, genetic engineering, and the environment. So we're going to be looking more broadly at the environment, but I will, of course, zoom in on humans towards the end of this. Just thinking about the big, it's not just going to be a bioethics talk. I want to basically raise the challenge of the rapid advance of this new field, how it seems to have caught a huge number of people unawares, and the big questions that society needs to ask about the renewed urgency of the place of humans in terms of the biosphere, the natural world, the created order, and what we can and can't do, what we should and shouldn't do. This has gained so much momentum recently, and I do want to sort of shed light on it. Just a little more on my own background. I did do a theology degree and then a master's in science and religion. My undergraduate was in genetics. I did a year of immunology, then I did a master's in molecular biology, and before doing PhDs in history and philosophy of science, and then in biomedical science at Reading. I've got some qualifications in education as well, but I'm normally dealing with people a lot younger than everyone in this room. No offense. Not to be an offense. So thank you very much for having me here. Right. Let's start with the obvious. What is SynBio? I'm just going to refer to it as SynBio from now on rather than synthetic biology. It's the human-directed and instigated utilization of biological organisms. It's us utilizing the natural world and other organisms, more often than not microorganisms, to produce the ends that we want. Utilizing them to, we take them and we get them to produce what we want, when we want it, how we want it. And this umbrella term oversees all aspects of genetic engineering. Give me a nod if you've heard of CRISPR-Cas9. Yes, give me a nod if you've heard of base editing or prime editing. Not so much, but someone might get a Nobel for them down the line as well. They're the newer tools than CRISPR. Everything from bioremediation to developing biofuels or new antimicrobials. You can also think of technologies such as golden rice, which I'll get into a little bit later, which you may be familiar with. But also how we can use SynBio to redress some serious climate issues. I think SynBio is actually our best tool in terms of dealing with climate change, but it doesn't often come up in conversation as the best route to addressing these. On the optimism side, and I am a science optimist, a tech optimist, I like to sort of think of myself as a futurist. You could see it as our best hope for not only climate crisis, but food crisis as well. But you can see it as our best chance of living off planet, if you want to go that far, and we'll touch on that a little later. It's our best tool for reducing waste. It's our best tool for dealing with pollution. It's our best tool at working towards medical equality. SynBio, I think, gives the best hope of all those things. But if you're a pessimist, and I fully understand if you are, you can start talking about Frankenstein's monster very, very quickly. And I appreciate that, certainly. It's you can talk about eugenics, is the other great fear. And that's not a word I use lightly. You also have unforeseen ecological disasters that could come from the best means bringing about the worst ends. And I fully appreciate that too. But this isn't about to go back in the box. It's not one of those technologies you can undo. It's so cheap now. It's so easy. It's so accurate. You can do a lot of it in a high school lab for a few dollars. It's a CRISPR world now, for better or for worse. It's not something we can undo. Give me a nod if you're familiar with Craig Venter. Craig Venter, of course, is very important in the race for the Human Genome Project. But was sort of one of the most famous popular scientists start developing synthetic cells, the creation of new genes. and a human-directed organization of cell and molecular biology. So that's what we're dealing with when I'm talking about SynBio here. Harnessing living organisms for our ends. This is such a booming, exciting field. We have departments of synthetic biology at the University of Reading, where I work. We also have them at Cambridge. Give me a nod if you're familiar with the term directed evolution. Again, something that would maybe have been fantasy or a prospective discussion a couple of decades ago, but it's now a real-world application where you are steering and accelerating evolution to bring about desired outcomes. The question here, obviously, if we can improve nature through directed evolution, is that something we should do? Jennifer Doudna won the Nobel Prize in 2020 for CRISPR-Cas9, and I really recommend her book, A Crack in Creation. It's one of those books that I gave to all my family members and friends for various Mother's Days, Father's Days, and birthdays, and things like that. Jennifer Doudna talks about how her attitude to how we should use gene editing in nature changed when at first it was all very frightening discussions of a new eugenics and Frankenstein's monster, but then being confronted with the question, if you have in your hand the power to do good, make things better, or alleviate suffering, and then you don't use it, at what point is that more unethical than its use in the first place? Which is an interesting way of reframing the question. I was at a conference at the University of Reading yesterday where a really amazing scientist was talking about addressing the growing mosquito problem in the UK simply by digging ditches deeper so that predators could then inhabit them. You know, rather than just being shallow enough for the mosquitoes to thrive, you build them deeper so that predators can then thrive there, and then they balance it out by eating the mosquito larvae or the mosquitoes themselves. This seems like a very simple human application. We're just changing the habitat slightly to address what could be a serious problem when you have mosquitoes now bringing things like the West Nile virus into the UK. But that is a human implementation. On the other hand, you have something as controversial as gene drive. Give me a nod if you're familiar with gene drives. Yeah, gene drives is where you use gene editing to manipulate populations of mosquitoes, then release them, and they outcompete the males, and you see a huge drop in malaria, which is an incredible thing to do, but it's very controversial. You always find large-scale protests when you're dealing with gene drive populations like in Brazil and other parts of South America. They're both human instigated efforts to address a serious health problem, but they're both one of them is controversial, the other one is not. So at what point are we improving nature in a way that is beyond pale? Is a fair question. I'm a big fan of Lord Martin Rees. Lord Martin Rees, who's a world famous astrophysicist, he's the Astronomer Royal in the UK, and he was very helpful to me in my PhD. And Martin Rees has sort of taken this a bit further into some of his popular books, talking about what one could do for the natural world with CRISPR-Cas9 and gene editing technology and synthetic biology. He supposes that it's not all that far away where we can CRISPR humans so that we can live on other planets. You know, if the moon and Mars and beyond is our destiny, if it's our destiny to colonize the solar system, you should use CRISPR-Cas9 to alleviate the suffering that comes from living in extreme environments of temperature, radiation, and all of those things. But at some point, then you would essentially be dealing with a different species of human. Now, I know the definition of a species is very fuzzy in philosophy of science, but at what point are you now not dealing with humans, but CRISPR humans? And all the associated questions of, do we have the right to colonize new planets? So I'll get to that in a second. But this is the idea of the invasive species problem. On the one hand, you're dealing with invasive species moving into new habitats and causing sometimes quite catastrophic change in new habitats. And that's because of things like climate change. And now you have a tool in SynBio to address that. On the other hand, we are an invasive species. And how do we deal with that using the tools of synthetic biology? Before I go any further, I think it might be helpful to flag up some of the key landmarks. This is Jennifer Doudna and Emmanuelle Charpentier, two remarkable women who were the first ever women to share a Nobel Prize in 2020 for CRISPR-Cas9. But I think SynBio, really, to be fair, you've got to go back to the birth of agriculture. Us harnessing and getting to grips with and shaping the natural world to suit our ends. You know, we were hunter-gatherers, we were beachcombers. The birth of agriculture, in many ways, is the birth of synthetic biology in terms of us shaping nature for our own ends. Then the selective breeding of animals, and Darwin talks about this at length in The Origin of Species in 1859. Of course, you can't talk about the history of synthetic biology without mentioning eugenics, most famously by the Nazis in the run-up to and then World War II. In the 1960s and 70s, following the first revolution in molecular biology, with the elucidation of the structure of DNA, you have DNA recombinant technology. Give me a nod if you're familiar with the first ISSR Conference. Very, very famous conference gathering in the history of science in 1975, where the genetics community invited in ethicists, philosophers, journalists, academics, theologians, people in government policy, lawyers, to the table at ISSR for a big conversation about, we have this new technology, what do we do? And it was only a few years ago now that ISSR 2 happened, symbolically, after the CRISPR-Cas9 revolution. Again, these should be much better known than they are. But again, the genetics community, yes, it has eugenics in its past, but it also has these moments where these new technologies have advanced and the genetics community has invited in people from the humanities and the legal side to discuss this and say, we have this tool, how should we use it? Of course, cloning follows shortly afterwards. Got to mention Fred Sanger, my science hero, double Nobel, won the Nobel Prize in 1958 for sequencing the first protein. Its amino acid sequence, and in 1980 for his development of Sanger sequencing for sequencing DNA. Obviously, a huge technological leap that birthed the Human Genome Project. Dolly the sheep, you're all familiar with Dolly the sheep, probably the most famous cloned animal. A lot of students sometimes think that Dolly is the only one of her kind. Like, no, Dolly was just famous. Cloning has gone a lot further than Dolly. Bioremediation, we have a catastrophic oil spill, and we utilize microorganisms to eat it up and clean it up for us. Also, think about Chernobyl and the area after the nuclear radiation leak. Harnessing microorganisms to detoxify an environment, that's such a fantastic example of SynBio and what we can do with it. The Human Genome Project obviously took 13 years, 1990 to 2003, about 50 countries, over 4 billion. You can now do it with a handheld device in the field for £500 in a couple of hours. That's how much the world has changed. Then in terms of our editing technology, Talons was the first venture into it, not that accurate and quite clumsy, but a great first leap. RNA interference, Mello and Fire shared the Nobel Prize for that back in 2006, CRISPR-Cas9. And then golden rice, I think, is a terrific example of using this technology to address serious problems with nature in a great way. One way I like to highlight just how much biology is accelerating today with students is just look back at the last 10 years of Nobel Prizes. I think, and some people disagree, but I do really think I'm right about this. I think biology is in a revolutionary period. It's in a data revolution. It's in its biggest boom since the 1950s, I think. 10 years ago, the Nobel Prize was shared between three incredible people for elucidating the methods of DNA repair. And I work in DNA repair, so those guys are heroes. 2017, the Nobel Prize was awarded for cryo-EM, which was our main method of structural biology at Birkbeck when I was there. And that means we can visualize macromolecules in solution in a more dynamic way than crystallography, and now to the same level of resolution. Cancer immunotherapy, which was called a fairy tale and a myth back when I was an undergraduate. A professor said the fastest way to take your career nowhere, you want it to go to a dead end, get into cancer immunotherapy. And then just a few years later, the Nobel Prize got awarded for it. And now it's a booming field. It's everything we've been waiting for. CRISPR-Cas9 2020. 2022, Svante Pääbo, who's our guest at the Genetic Society Conference this year, won the Nobel Prize for sequencing the Neanderthal genome. That's the world we live in now. Not quite Jurassic Park, but not a million miles away either. And then in December, the Nobel Prize was awarded for AlphaFold, a tool I use every week. And at Reading, we develop tools to compete with AlphaFold, and some of them succeed better than AlphaFold at various aspects of the structure of macromolecules. But this is such an incredible 10 years. But what's really, really important about this is a lot of these crossover and inform one another, creating this snowball effect. And this is compounded by the fact that gene sequencing and genomics is now so fast, so accurate. What took 13 years and 4 billion in 50 countries can now be done with a handheld device in the field. Kate Rubins, the NASA astronaut in 2016, sequenced the genomes of all the microorganisms on the International Space Station with a handheld device. That's the world we live in now. Absolutely extraordinary. What took 13 years, a painstakingly slow process, can now be done in space with the Minion, which is about the same size as this. So what that's doing is creating a data revolution that's accelerating this snowball effect. So SynBio, which was this futuristic sort of mythical idea about the kinds of things the future could look like, is now here and happening. And I think regrettably, people are so distracted by the AI revolution, they've missed what's happening in biology. I'd say even at science and religion gatherings, everyone's talking about AI. Like, have you not noticed what's happening in biology? The world is changing and shaping in a revolutionary way that rivals that of the 1950s. And Christians have not got on board with it so much. Even philosophers of biology have not quite got there in the same way they have with AI. And we need to be sure. I just released a paper that's about to come out in Science and Christian Belief about the three-tier problem of science communication that is compounding this issue when we think about SynBio and the natural world, both for better and for worse. On the one hand, you have the science, which is accelerating. And you have a big gap between where the science is and the ability and number of skilled science communicators to disseminate this to the general public, as well as to the academics. The gap between what the public knows and where the technology is huge, and it's growing. But then you also have a gap between that and the theologians and philosophers and the ethicists to reflect on it and take time on it and process this and meaningfully engage with this as well, which is a challenge. Let's go to a great example of SynBio, which is golden rice. Places in the world where there's a massive vitamin A deficiency causing really bad health outcomes. Golden rice, you edit this rice so the vitamin A content is so much higher. And there you go. You have a massive nutritional and vitamin deficiency health problem in large parts of the world addressed through the use of SynBio. It's a great triumph. Over in the Western world, more affluent parts, we're really massive on our vitamins and supplements. And here you have SynBio kind of leveling the playing field a little bit better. This is a great example. I'm a big fan of George Church, who is the geneticist behind the idea of bringing back the woolly mammoth. Anyone seen this in De-Extinction? This is a more of a kind of crazy out there idea, but George Church, I'd not put it past him to do it. The closest homologue to the woolly mammoth is the Indian elephant. And so you use, much like Pablo did with the Neanderthal DNA, we have the genome of the woolly mammoth. You engineer a woolly mammoth Indian elephant hybrid. And the rationale behind this is climate change. Basically, you bring the woolly mammoths and you have them wandering over the Siberian steppes, pumping down the permafrost like they used to in the previous ice age. And under there, you've got huge amounts of methane that if climate change took off, unfortunately, would be released into the atmosphere. A serious problem, methane, obviously a massive climate disaster would come from that. So, de-extinction, bring back woolly mammoths, let them wander over the steeps, tread down the permafrost, keep the methane down. Here you've got a SynBio idea that's guided by climate change, but it raises massive questions of the theology of de-extinction and what comes with that. And I had a debate with 𝐂onnect on Premier Unbelievable 2 months ago about this. I was making the case for, but 𝐂onnect was saying, well, wait a minute, you know, there are theological questions to ask about de-extinction. We can't just say, well, it would be better for the climate. You know, are we then going to say, well, extinction doesn't really matter because we can just sort of bring them back? What are the hang-ups going to come from that? A better one, though, is maybe the American chestnut. The American chestnut flourished, particularly along the east coast of the USA. It was very, very important for squirrels, bears, caterpillars in particular, but viral infection and deforestation almost completely eliminated it. But with genetic engineering, American chestnuts for fungal resistance, you could bring them back and see them flourish up and down the East Coast. I think that's something that people would say, well, wait a minute, yeah, actually that is something I could get behind a lot better. But we have questions to ask there. I want to highlight two thinkers, just as we look at this. During the time of the DNA recombinant revolution, the first is John Hick, who wrote this wonderful book called Biology and the Soul, which is a little treasure people haven't heard of. You can still get it, though, because it was printed by Cambridge University Press. You can probably get it for £2 on Amazon. But he gave the Eddington Memorial Lecture at a time, this was 3 years before Asilomar, actually, talking about what do the questions of recombinant DNA mean for us, so that looking back at this explosive birth of molecular biology as a discipline, what do they mean regarding our conception of human life in the context of molecular biology? Who are we in the world of molecular biology? And it's a real gem to pick up. The next is Arthur Peacock. In 1986, wrote a book called God and the New Biology. I have a spare copy here for anyone who would like it. Whoever asks the best question can have the prize. The new biology, I love that term because we need to do this again. Someone needs to write a new Peacock book today. God and the new biology. Now, Peacock was mostly writing in response to sociobiology by E.O. Wilson, also Jacques Monod's Chance and Necessity. He was asking big questions about what it means to be a special creation in an age of chance and sociobiology. But I love the phrase he uses. He talks about a new context for enrichment. He was looking at it in a positive way. How does the new biology enrich our understanding of who we are? And I'm calling for a new one, a new Peacock, a new Hick response today for one of you academics to come up with. Just like in the 1920s and the 1930s, a lot of philosophical physicists, many of whom were theists, were reflecting on how our knowledge of the universe was enriched by the new physics. We need to allow our theology of ourselves and of the natural world to be enriched by the new biology, and understand humanity and creation in this new context for enrichment. And I don't think we can wait, because I'm just going to give you this little warning here at the end. For those of you who saw this in the news 2 weeks ago, of the first CRISPR baby whose life was saved by the intervention of CRISPR-Cas9. Now, 𝐬𝐂onnect in 2019 edited a pair of unborn twins and was put in prison for it, fined, and his lab was shut down. He's since been released, given his lab back, and told to carry on his fine work because he edited a pair of unborn twins and the mother's consent was highly dubious. This child would have died if not given this CRISPR-Cas9 therapy. So in a sense, a lot of the ethical questions about the use of CRISPR on humans was skirted over because it was a life or death situation. But the thing is, in California, if you've got 50 grand, you can go to Genomic Prediction, take your IVF cells, and they have a checklist of about 200 questions you can ask. How would you like your baby? What color hair would you like? What color eyes would you like? How tall would you like them to be? What kind of muscle mass are we looking for? These genes are correlated with intelligence. We're talking about a genetic upper class for those who are rich here, who can give their children every single advantage they want. But it is the instinct of us as parents to want to give our children every advantage. So does it matter if it happens before or after birth? These are things that we can't really wait to reflect on anymore. But I'm out of time, so I'm going to stop there and take your questions. Thank you very much.
Thank you very much, Sam. You've got just over 5 minutes, so if you want to just field the questions yourself.
Sure. Yes, Richard.
That was super. Thank you very much. Two questions, really. The first one is, you mentioned a lot of the public space being gobbled up by artificial intelligence. With the super presentation you've given, a little bit more about why that's taken all the space, and this topic has not had quite so much of a high profile, although it's on some high profile.
Yeah.
Why that imbalance? And then the second part of it, really about, you mentioned about theologians being slow into this area. What can be done to encourage both theologians and indeed senior church leaders?
There's a couple of layers to that, and in my answers, I recognize an advanced disclaimer, some of them are going to be controversial. But I bet right now that there's probably 100 books on theology and AI for every one book on theology or biology in terms of the new biology. Yeah, the last 10, 15 years. Post-Human Genome Project, let's say. Partly it is, AI is a very worthy distraction. It is absolutely a worthy topic to take all the attention. It deserves the attention it's got, and it shouldn't be diminished. I'm a huge fan of AI in biology. I mean, AlphaFold is going to change drug discovery and medicine forever in ways that people don't even know. Part of the problem is people don't even know. The second thing is, I think when it comes to physics and computer science, the church is a lot more ready and happy to engage with that than it is with biology. Just historically, if a physicist wants to give a sermon, the vicar's probably going to say, yeah, brilliant, go ahead. And the church is going to say, oh, I can't wait. If a biologist wants to give a sermon, they're probably going to want to know what you're talking about first. It's just, unfortunately, the history of our disciplines. So that's part of it. But I do believe the biggest problem is science communication, both in terms of the distraction, but the gap in biology is particularly acute, science communication. This question, yes.
You said at the break earlier today that this is the best time to get cancer, and I understand what you mean by it. My question would be, what are the parameters of synthetic biology to be able to address things like fibrous dysplasia, ossific Asperger's, or other, Huntington's chorea, things that are intractable now? Are they addressable? And if so, what are the limits?
So I don't know all those diseases, but I do know Huntington's, because Huntington's is a great example of a clear genetic disease. Now, let me tell you diseases that have had an absolute revolution in the last 3 years because of CRISPR-Cas9. Sickle cell, beta thalassemia. You're talking about diseases where it's a single point mutation, like a single base pair change that's causing this cascade medical catastrophe. Huntington's is one of those. And I think Huntington's could go the way of smallpox with genetic engineering. It could be a disease that could be eradicated, either in terms of editing in the womb, editing through IVF, or maybe even editing afterwards if you can get the therapeutics right. Genetic diseases that were impossible or seemingly intractable are now not, because CRISPR is so cheap, accurate, and easy. So that landscape is going to change. There are things that are easy to predict. I think that's an easy one to predict. Everything else is no longer impossible. Who knows where it's going to go? Yeah.
Okay, thank you very much. Lisa Siderus is talking a little bit later, and I know I don't know what you're thinking through things like the woolly mammoth and de-extinction and that kind of thing. So there has been quite a lot of interest among theologians on de-extinction. I was also involved in the sort of working on CRISPR-Cas9 and the ethics thereof from organized through an ethics center in the US. And I think you're right that we hasn't been enough catching up. But my question has to do with maybe some of the pushback. For example, vitamin vitamin rice that you mentioned, that is also it's also possible, according to Vandana Shiva, to find that amount of vitamin in a natural rice. So is that kind of technology really necessary? So in other words, there's a certain resistance based on some of the cultures or indigenous peoples who perhaps feel like that this is kind of being imposed, from the West, as it were, and not taken into account what they really need or want, and the fact that there's naturally occurring one. So there are those sort of big issues as well. So what I'm saying is that some of the hype around these technologies and the public communication, I would caution against scientists making too sort of radical claims about the benefits without actually looking at some of the potential cultural or other risks associated with it. I think these are very real. So it's a similar situation happened with the Human Genome Project at the turn of the century, where there was an awful lot of hype about all the different diseases it could potentially solve, and then only a couple of them, one that was sort of directly linked with certain genes, were even sort of viable. So, I mean, how do you respond to that?
Yeah, plenty of that's fair enough. I know I have to be brief. It's before the Human Genome Project had finished, you didn't have talons or you didn't have RNA interference, those sorts of things. But let's just look at a couple of those, like DNA repair therapy and immunotherapy are the future of cancer treatment. I think we will get to a point where cutting it out, burning it or blasting it isn't as effective as DNA repair therapy like PARP inhibitors or cancer immunotherapy. It's just so quick and so new. But then you couple that with something like AlphaFold or cryo-EM. Like I said, these things they inform one another and are creating this snowball effect of opening up possibilities that weren't there before. Yes, there's always hype. I think with CRISPR, and not just CRISPR, prime editing and base editing, you don't even need to induce a double-strand break. You can just change a base without having to break the DNA helix and then use non-homologous end joining or homologous recombination to then put it back together. So it's even less invasive than those. I just think the hype is real with that. If the world of cancer then becomes, I sequence your genome because it's so cheap, easy, and quick, and I couldn't do that before, then I take a biopsy of your tumor and I sequence the tumor, get the tumor signature, then I take my immunotherapy drugs, and I use gene editing to get the CAR-T cells uniquely specified to your tumor and your physiology. You take them orally or an injection rather than having to blast yourself with radiation or chemicals. And not only does the cancer go away, even if you've been sent home to die, but the cancer then doesn't come back because your immune system is primed against it. I mean, that's the world we're looking at in the next 10 years. So that's not hype, that's technologically possible. It's just too expensive right now. But the costs will come down as more biotechs get involved and more research happens. So it's absolutely fair enough to look at that in the past. I just think now we should probably give the devil its due. That's a horrible way of phrasing it at a conference like this. But and play safe rather than sorry. Thank you.
Okay, on that note, we'll end it there. Thank you very much for doing that.
Thank you so much for watching. Don't forget to like and subscribe. Stay tuned for more amazing conversations with incredible researchers who are changing the world we live in and driving humanity forward. Feel free to check out my Instagram at polymath_Sam or go to my website at www.sam-mckee.co.uk.